Okay, I'll try and finish the second part of this talk in about 20 minutes. Okay, so this is where we left off. Blood glucose levels. Here we see the normal range between 60 to 90 milligrams per deciliter. If it drops below this down to 60 to 40-ish, we see subtle neurological signs, hunger, obviously, but some lightheadedness, confusion perhaps, sweating, trembling, shaking, because you're hungry. And then if it drops down below 40-ish, lethargy. So what's like couch potato-ish, you cannot move, or you can move, but you don't want to, right? Lethargy. Convulsions even when it gets more serious. So, and then going into a coma at this stage. And then down below 20 leads to permanent brain damage over a prolonged period of time, eventually death. So the hormones that regulate this, almost all of them, blood glucose levels, we have touched on: insulin, glucagon, epinephrine. Cortisol we haven't, and we will just mention briefly. I'm not sure if it's in this part or in another part. Hormones in general, this stage you don't really need any more introduction to them. Glucagon, insulin, epinephrine and cortisol, not sure. But these three are endocrine signaling over a long distance. I think cortisol might be more localized, so a paracrine signal, but we'll see when we get to that. Types and mode of action of each class can be different. Peptide, the type is a peptide, is insulin, glucagon. What we see here, steroid hormones, like cortisol would be a steroid hormone. It has a nuclear receptor instead of transmembrane, plasma membrane receptors like G protein coupled receptors or receptor tyrosine kinases. So hormones are essential to multicellular animal and plant cell development and communication. They're needed for regulation of metabolism, regulation of food intake, sexual differentiation, and blood supply. And we are just focusing on the metabolism ones in this MOL200. Peptide hormones, we just mentioned this very briefly. Insulin is formed as a pre-insulin peptide, which needs to be processed. The signal sequence, if you haven't heard, a signal sequence on a protein means that it's destined to be secreted out of the cell. This has to be cleaved off at some stage. And then proinsulin gets some disulfide bonding to give it a more rigid shape. And then the mature insulin cleaves off this C-peptide, which is an metabolically inactive peptide, to give us the ligand insulin that will bind to the insulin receptors, stimulating them. The C-peptide is actually what's used in diagnosing the amount of insulin released. If that's a test that you have taken before, they look for this. They don't look directly at the amount of insulin, they instead detect the amount of C-peptide. When it comes to insulin production, insulin and glucagon and somatostatin, there are these three cell types: alpha, beta, delta cells found in these clusters of cells or islets of cells called the islets of Langerhans inside the pancreas. Right? The pancreas has two different types of flow through it. It has blood flow, which is important for us when it comes to the secretion of hormones, because the blood vessels penetrate these islets of Langerhans so that the cells, the alpha, beta, delta cells, can secrete directly into the bloodstream. There are other tubes. These are ducts that can take enzymes important for the digestive system from the other cells of the pancreas that we don't really talk about. So these pancreatic duct cells. So for us, when it comes to hormones, they're not interesting, but of course for food digestion, they're very important. So insulin and glucagon are the key enzyme hormones we have been talking about, and this is just to show us schematically how they regulate blood glucose. So pancreas, the liver, blood glucose levels in balance, homeostasis, about 90 milligrams per deciliter, let's say. Then it goes out of balance this way. We have decreasing blood sugar. This leads to stimulation of the pancreas, the alpha cells, which will release glucagon, which activates G protein coupled receptors in the liver, triggering the breakdown of glycogen to glucose. Glucose is released from the liver, blood glucose goes up, and in this case, and in cases of eating a lot of food, we get an imbalance with this rising blood glucose levels goes too high. This is sensed by the beta cells in the islets of Langerhans in the pancreas, which secrete insulin in response, and then this will bind to tissues that express the insulin receptor. These will be so-called insulin sensitive tissues, and these tissues will then be stimulated to uptake glucose. Typically, the mechanism we've talked about is the transport of GLUT4 containing vesicles to the cell surface. This will cause blood glucose to fall back towards the normal range or below, and then we go back around. So these things are constantly going to keep a balance, right? I hope that's clear. How this looks after eating a meal, I think we looked at this before already. So we just go through quickly. So here is like you wake up, you have a meal, and your blood glucose goes up. During nighttime it was pretty low, goes up, you get a spike in insulin, and then this causes your blood glucose to go down. You wouldn't see this decrease if you didn't have this insulin spike. And then it follows through. Okay, the day you have another meal, have another spike in insulin to keep the glucose down. Insulin drops, and you have another meal. Glucose goes up, insulin rises. And the opposite is true for glucagon, even though the spikes aren't as severe, but during periods of, here's the biggest spike, like in glucagon, it's starvation, semi-starvation period between the noon and evening meal, right, where blood glucose was low, glucagon goes up to try and get it up again. And the goal of this up and down glucose and glucagon is to try and keep blood glucose as balanced as possible. This we have seen already, insulin stimulation of pi3 kinase, causing the generation of pt2, so phosphorylation of phosphoinositol 4,5 bisphosphate to phosphoinositol 3,4,5 triphosphate, which then this lipid, phosphorylated lipid, is recognized by domains on these proteins PTK1 and AKT, which bind to it. PTK1 phosphorylates AKT, which indirectly leads to the trafficking of GLUT4 receptors to the cell surface, allowing glucose to come in. And following release of insulin, within 5 minutes in insulin responsive tissues, we see an up to 30-fold increase in glucose import. So it's a rapid response. So what we haven't talked about is how these cells, the insulin secreting cells, actually detect rising glucose. And it's a little bit indirect, but we can go through it anyway. So actually, these pancreatic beta cells, they express the GLUT2 glucose transporter, similarly to the liver cells, which is a bidirectional transporter, transporting glucose down a glucose concentration gradient. We get glucose import into the cell. When it's in the blood, we will then, remember we had this blood flow through the islets of Langerhans, so the pancreatic beta cells get exposed to this glucose, blood glucose rises. Similarly to the liver, pancreatic beta cells have hexokinase 4, the glucokinase, converting glucose to glucose 6-phosphate. And it enters into glycolysis, the citric acid cycle, oxidative phosphorylation to give us an increase in ATP. This is where things get interesting. ATP binds to an ATP-gated potassium channel, and ATP will inhibit this ATP-gated potassium channel. And normally when it is active, it pumps potassium from inside the cell to the outside of the cell, creating a polarized membrane with increased charge on the outside compared to the inside. But when ATP binds, this efflux export of potassium is blocked, so the cell depolarizes. So this charge is lost. This depolarization of the membrane is sensed by the voltage-dependent calcium channel, which allows influx of calcium. This is then thought to be the increase in calcium intracellularly is boosted by release of calcium from the ER. We get an increase of calcium overall, which triggers release of insulin granules from the cell into the bloodstream. Remember, this guy's the beta cells are surrounded by the bloodstream. Just have a look at a picture of a pancreatic beta cell on EM, very large nucleus here, and we see then these granules. And if you were to zoom in, you would see then these granules have these funny shapes inside them. That's actually packed insulin. So we have the processing of insulin that we mentioned, and then it is these final processed ligands are processed or packed together into an insulin hexamer on these granules sitting waiting to be stimulated. And we see that the plasma membrane or the vesicle membrane of these granules is pretty complex with a lot of different kind of channels and stuff on here. So the ones that I'm familiar with mostly are like these RABs and snares, synaptogenins. These are involved in vesicle trafficking and fusion with the membrane. So you have to think that this vesicle has to go and fuse with the membrane, and it will open the membrane, and all its contents will be released out into the outside. But the membrane itself will actually be left on the cell surface, right? And maybe this is then a way to put these receptors also on the cell surface, and these are many channels and stuff like that. So they may not be completely functional when it comes to insulin secretion. They may not have a role there. They may be just passengers getting to the membrane. That said, this is just one way in which this ATP depolarization of the membrane is just one way in which the insulin has been shown to be triggered to be released specifically by this increase in glucose. There are other ways, and some we will touch on. Therefore, these may play a role there, but you don't need to know, and I don't know. This is the ATP-gated potassium channels. This is in the book, but this year we're not really going to look at it, so it's only for those that are interested. There are a set of drugs available that can open or close the channels, triggering this depolarization of the membrane and therefore triggering release of insulin. And these can be useful in certain cases of diabetes. So a bit more information here. Again, just press pause if you're interested. Again, just press pause if you're interested. Yeah, back on. So I mentioned there was more than one way in which these insulin granules could be secreted, and I think that it's quite an interesting way, but I don't know what you think. So there's this pre-absorptive cephalic phase of insulin secretion, which, I mean, we've been showing you this. Look, you eat and you get a spike. But in truth, it looks like this. What happens is that you look at food or smell or taste food and you get a spike. And this is called the cephalic phase of insulin secretion. So it lasts for about 10 minutes. Just by looking at the food, you know, get this secretion of insulin. And this is kind of to prepare the body to get rid of that glucose, to internalize that glucose as quickly as possible, even if it's not coming. You know, of course, you can trick your body. And this is a behavioral kind of response, right? You have this parasympathetic nervous system that is coupled to the pancreas and can actually stimulate these cells. That's why I was saying maybe some of these receptors on the surface of the vesicles, but inside the cell, maybe they're somehow downstream of this parasympathetic response. So it's not like it's not the sympathetic, it's the uncontrollable response, but one that can be conditioned, which is quite interesting as well, if you know what conditioning is. But then I asked later or earlier in our talk, what happens when you drink Diet Coke and stuff like that, you know, things with artificial sweeteners? Do you get a spike in insulin? by ingesting these very sweet things. And yes, you do, of course you do. You actually get it just by looking at the can almost, right? And also opening it and smelling it, you'll get this release of insulin. But maybe over time you won't, if you, from just looking at it, because if you don't open it every time. But every time you taste it, you taste something sweet on your tongue. You get taste buds in your tongue. Obviously, are connected to your brain, and that will be connected to behavioral centers in your brain that will trigger hunger, or in this case, even just this parasympathetic response to secrete insulin to prepare for food coming in. So would it affect blood glucose levels? For example, just looking at a can of Coke, would it affect your blood glucose levels? And probably you guys are not so interested in these things. I just put it up here. It doesn't seem like it, right? And then this is for those that are interested when it comes to this sort of anticipatory behavior. So but just explaining the experiment very quickly. This is not on the test. I forgot to write it. You have, in this case, we have the unconditioned sugar. So you give sugar in water to a rat, I think it was, and then you measure the insulin secretion. It goes up when you give them sugar and continues up when it starts absorbing from the gut and actually reaches through the bloodstream, reaches the pancreas. Here you give the same looking solution, but it's nothing sweet in it. It's actually salt. And then you get this, just the conditional response that it goes up. I'm not sure why it goes up again. I haven't read this very well. But this is the conditional response. And how do you know it's a conditional response? Because you can take it away. You can actually give them the sugar with poison in it that tastes terrible, and you don't get this response. You have to train them for that then, right? So teach them that they're getting something horrible, and then they won't have this same response, that they'll secrete this pre-absorptive cephalic phase. Insulin secretion will be missing. But when it reaches the gut, this poisoned sugar, you still get increase in insulin then, right? Okay. And if you want to interested in things like that, Google Pavlov's dog and get some idea of how these things work. So just summarizing this part here, the sight and smell of food might be as bad as drinking a Diet Coke. And if you're taking like a Diet Coke with food, should you care? Not in terms of your insulin levels, I would say, or blood glucose levels, but there are some disturbing reports that the sweeteners for some of these drinks, for example, they react differently in the gut when it comes to metabolism of these sugars in the gut by bacteria. So often these sweeteners, they have no calories because we can't break them down and extract energy out of them, right? But there can be bacteria in our gut that manage that, and if we feed them the thing they like, they can expand in their population, and this can have a negative effect on your gut flora and even cause inflammation in the gut, for example. And this is something that we take up in a later talk because it is in the end of the chapter. So this figure summarizes the effect that insulin signals have in response to high blood glucose. They stimulate glucose uptake. So from the pancreas, insulin secreted to the brain, adipose tissue, and muscle, upregulating the expression of glucose receptors to import glucose. And yeah, in the muscle. Yeah. Then in the liver, it will encourage the conversion of glucose to glycogen. That's it, I think. You can read through this yourself. There's a lot of things that we covered step by step earlier on. Try and remind yourself of these steps here by reading through this table. So I have a question here, actually. Considering what we know now about the muscle and the liver in terms of glucose uptake and storage as glycogen, after a carbohydrate-rich meal and a release of insulin, which of those tissues, the liver or the muscle, is responsible for taking up the majority of the glucose from the blood. I want you to have a think about it and reason why. Why do you think that? Don't just guess one or the other. Glucagon is also a peptide hormone, small 29 amino acid polypeptide, synthesized and secreted from the alpha cells in the islets of Langerhans. It has a constant production, but it's suppressed or regulated by amylin, a peptide hormone co-secreted with insulin from the pancreatic beta cells. When plasma glucose levels drop, you get reduced amylin secretion, reduced insulin, and then this allows for glucagon secretion. So here we see actually labeling of islets of Langerhans, where we label, so all the DNA is labeled with a stain in blue, and then in green we have the the beta cells in green, and the glucagon expressing alpha cells in red. So if this is antibodies against glucagon or insulin, I'm not entirely sure. This is just to show you the actual how it corresponds here, these models. Glucagon regulation, secretion of glucagon is stimulated by low blood sugar, by epinephrine, and then alanine as well, and some other things. And alanine, then remember we talked about that when the muscle is, or when the body is starving and starts breaking down proteins in the muscle, we get it shifting around the body as alanine to be converted back to glucose by the liver, and this alanine is sensed then in the alpha cells and triggering increased secretion of glucagon. Glucagon secretion is inhibited by somatostatin, a hormone. I think we talk about it later. Amylin, we just mentioned, secreted together with insulin. Insulin itself, and free fatty acids, when they're in the blood. So if you have free fatty acids, I guess that could be telling you you've just had a very rich meal, right? And then we have the same figure almost as before, but with glucagon, and the primary target of glucagon we can consider to be the liver, changing its metabolism, right? Encouraging the breakdown of glycogen to glucose 6-phosphate, and then that can be transformed to glucose in the liver and exported to supply the brain or muscles. We get the change from glycolysis to gluconeogenesis, which is glucagon regulated, and then production of ketone bodies from fatty acids, and then the breakdown of amino acids, proteins and amino acids to pyruvate for gluconeogenesis, all to supply the brain primarily with glucose. And there is a table here summarizing many of the enzymes that we talked about earlier on when we're discussing regulation of glycolysis, gluconeogenesis, and glycogen metabolism. Then just to revisit this figure, the liver serving as a metabolic hub in the body, coordinating the exchange of metabolites between major tissues and organs. And the pancreas isn't on it. Damn it. But all of the processes we have talked about are shown here. Okay, that brings us to the end of the second part of the first lecture. We just went quickly through that part there. A lot of things we've kind of covered before, and we're just trying to put things together now in a cohesive way. Hopefully it's working right. Before I go through this learning goals things, I want to say please participate in the poll, even if you don't have questions, and just to tell us that you're sort of keeping up to date and understanding. You can just write yes somewhere right there. I want to know how many people are finding this method of teaching useful, right? Where you have this opportunity to ask questions via this form, followed up with an opportunity to ask questions at a live Q&A, where we also have the opportunity to answer your questions in a prepared way. If you think that that is a good thing, please, you don't have to ask a question in the poll, just write yes. So we know how many people are getting use out of it, and we can compare it to how it was with live lectures, when basically we would have only between 0 and 1 question, let's be honest. Okay, so today's learning goals, we covered tissue-specific metabolism and how the organs divide labor up within the body and do accomplish different tasks. They work together. The liver is totally exploited by other tissues. That's the way it is, you know. The other tissues keep the liver alive as well, right? And get him from A to B so he can get food and drink and whatnot. So we looked at metabolism of sugars, amino acids, and lipids in the liver, adipose tissues, muscle, brain, and blood, and kidneys. And we discussed a little bit about their physiology. And then we spent the last part of the talk just discussing the regulation of secretion of insulin and glucagon and how they affect the different tissues, which is stuff we've covered in more detail in earlier lectures. We're just trying to pull things together now. The next lecture will kind of continue on from here, looking at the effects of fasting and starvation on the body organs and the different types of metabolites. Okay, thank you. Bye.